background is going to enlighten us on the effects of nutritional and pharmacologic interventions that extend rodent longevity. After earning a bachelor's degree in architecture from the University of Southern California in Los Angeles, he received a master's in terrestrial ecology before finally entering the research world of aging, completing his PhD in 1981. He began his career in academia at McGill University before assuming a faculty position at the University of Texas Health Science Center at San Antonio in the Department of Physiology. He is currently working as the co-director of the Intervention Testing Program at the Barshop Institute for Longevity and Aging Studies, and as the director of the Aging Animal and Longevity Assessment Corps of the Shock Center for Biological Research. He is also currently serving as president of the American Aging Association, which is the only scientific society in the States devoted solely to advancing research on the basic biology of aging.
Dr. Nelson's most recent studies have sought to uncover the mechanisms of dietary restriction on longevity in inbred mouse strains. By monitoring genetic changes, he has identified different physiological and molecular changes believed to play a part in the extension of lifespan via diet restriction. Dr. Nelson has also recently looked into the effectiveness of rapamycin, an mTOR kinase inhibitor, in increasing lifespan and the metabolic differences between diet restriction and treatment of mice with rapamycin. Please give a warm welcome to Dr. James Nelson.
Well, can you all hear me? Okay, thanks very much for that very thoughtful and thorough introduction. I really appreciate that, and I'm very happy to be here. One of yours, your probably favorite teachers and professors here, Rosemary, invited me to come. And one of the things she said was she told me there would be a lot of students here, and I think she asked me to give you not necessarily a classic seminar on my science, but also to try to give you some insights into what I think are the big questions for those of us who are interested in this process called aging, and how we can go about studying it, and what approaches we can take, and also some of the problems that we're faced with in trying to study this problem in a scientific way. I mean, aging, when you think of it, and I'll show you some pictures in a minute, is easy to think you know what it is. I mean, when you look at somebody, you can say, oh, he or she is getting old, or they're very old. But if you try to quantify it in some way so that you can approach it scientifically, it's quite a challenge to do. It's sort of like, when you think about what if we wanted to study the biology of love. We all know what love is, but how are we going to quantitate it and study it? Okay, so keep that in mind, and hopefully some of you will become interested in this field and can make progress towards understanding this really big problem. So I want to begin by and feel free to interrupt me anytime that you like. If we don't get through everything, the most important thing is to keep interactive and answer questions and see where those questions may lead us. But before I begin, I want to introduce some people who are key to the work I'm going to be talking about. And this is a picture of the Barshop Institute, which, by the way, is one of the few freestanding independent institutes that's devoted to the biology of aging. When I started, you may have heard that I have a rather serendipitous and circuitous trajectory in my career. As they pointed out, I was studying terrestrial ecology back in the 70s when this young professor from Rockefeller University came to USC where I was studying, and was actually planning to leave because I'd been there for all of my academic training, wanted to get away. He talked about this field called aging, and back in the 70s there were probably 5 scientists in the whole United States who were studying aging. His name was, and still is, Tuck Finch, and he was so seductive in his discussion of this challenging problem that I fell for the science and started working in his lab and joined it, never realizing that some 35 years later or so, the field would have made so much progress, but it's got a long ways to go. So anyway, like I said, this is now one of the few places where there's a group of people that are strictly devoted to searching for the mechanisms of aging. And these two people on the top are very instrumental in the intervention testing program, which was mentioned at the start. Dr. Strong heads the program at the Barshop, and Dr. Sharp is one of the people who, like you, could just come up with an idea and submit it to this program if you had an idea for something that might slow down aging, some nutraceutical, for example, or some drug. And Dr. Sharp had an idea that was Rapamycin, and we'll talk about that quite a bit during the talk. These people I also want to introduce. They work in the basement of this building, and they run our animal core. And that's a 365-day-a-year project, because one of the things we measure is death and what day the animal dies. And so you have to be there to identify when that happens every day of the year. But they do an awful lot of the hard work that's involved in studying aging in mice. And then a couple other people I want to introduce on another project. These people were involved in the dietary restriction study that also the two women who introduced me talked about. And Chen Yu Lao, who's now graduated and is at the Buck Institute, was my graduate student. He was primarily responsible for the work that we'll briefly talk about today. And the two people below are the brains in a way behind this work. They were the ones who came up with the idea of using genetic diversity to study aging and food restriction. Okay, so what is aging? I already alluded to this. We know what it looks like, but really to describe it in a way that you can study it and test hypotheses about what influences it biologically is a big problem. If you just think about it and ask yourself personally, how could I study this? It's quite a challenge. And then even if you know how to define it, and we have some ways, fortunately, of defining it, which are incomplete but useful. How can we determine the mechanisms responsible for it? And so I'm going to talk about a couple approaches on that soon. But so here's one definition of aging that I think is one of the most quantifiable, but it's also one of the hardest to get because it requires such large sample sizes. We say aging is that biological process that leads to the exponentially increasing incidence of disability, disease, and death with advancing chronological age. So, for example, death by all causes, shown in the left panel, has an exponential rise. And any other cause of death, heart disease and cancer being the two major causes of death in the Western world, also increase exponentially. There's a little change with cancer. Seems to sort of tail off after the age of 80, 90, or 100 or so. So that's a little bit of a blip. But anything else that you can think of that kills has this exponential with age killing strength. In fact, age-specific mortality is indicated in that sentence below by death by any cause. And that could be walking across the street and getting hit by a car, or having a third-degree burn. After you're 30 years old or so, the likelihood of you dying from getting hit by a car getting hit by a car, first of all, and then dying from the accident or suffering from that third-degree burn, the likelihood of dying from it doubles every 8 years of life after you're 30 or so. Okay, so it's a compounding interest problem, one of those compounding interest problems that we'd like to avoid. But clearly, this shows a way to measure it, because if you think about plotting this on a semi-log scale, this rate of increase, if you could change the mortality rate doubling time to every 16 years by some way, you could definitely affect the aging process, because you would affect all of these diseases at the same time. We're pretty sure about that. Okay, here's another way of looking at it, risk factors. This is somewhat, I would say not totally quantitative, but it just makes the point. We all know risk factors for cancer, and I was just talking with one of the colleagues here about preventing cancer, and that's something that many of us would like to put more of our resources into. But we do know that tobacco and alcohol and diet and possibly infectious experiences early in life are risk factors for many forms of cancer, but they all pale by comparison to aging because of that exponential increase in risk that increases every 8 years, doubles every 8 years from 30 onward. So the idea here is, from a prevention standpoint, if we could slow down aging, we're pretty sure, and we have animal models to suggest this, evidence-based, that we could have a big impact, a bigger impact on cancer than if we quit all the tobacco use in the United States, for example. But the problem is how to do that. But aging is a lot more, and here what I'm doing now is building up a case for how to define this thing called aging. Aging is more than just this increased risk of disease and increased risk of dying from disease, as evidenced by this one example. Here's a guy who's a very fast runner when he was young, and he's still fast as he's older, as you can see. But as you can see, as early as 30 to 40 years of age, average foot speed is starting to decrease. This has, to a large extent, nothing to do with diseases of aging. It has to do with this mystery, largely still unexplained, biologically speaking, about those processes that cause this decline in function with advancing age. And so there are many things that change with age that are not diseases of aging. Related to this is muscle strength, the rate at which nerve impulses are transmitted from your brain down to your calf, for example. All of these things slowly decline with age. And one of the questions is whether or not the processes that regulate that there's a single process that regulates it, or whether it's multiple factors that are involved. So these are, just some other examples of deteriorative changes with age that occur and have nothing or very little, as far as we know, at least in their early onsets, with those diseases of aging we talked about before. Anybody have any questions? Okay. So this is aging, and so as you can see, how to define it in a way that you can probe it becomes quite a challenge. Okay, so what causes it? There have been many hypotheses raised, and there's a lot of philosophizing, you might say, in the aging field. There has been, for what the underlying causes are. And these are just some of the hypothesized causes. Oxidative damage has been a biggie. There's this aging society that I told you about that I'm heading up right now was founded by a guy named Harmon, who put forth the free radical theory of aging. And he started this society and held on to it very tightly. It was a very family, mom and pop run kind of organization that he clung to, because the big society, the Gerontological Society of America at the time, they had a disagreement about what was important in biological sciences. GSA, Gerontological Society of America, is a big society that encompasses all aspects of aging, from social policy, public policy, to psychology, to medical geriatrics, and so on and so forth. The biological part is very small. And so he broke off from them because he really, I mean, he's a true believer. He truly believed, you know, and this is when you have to be careful, because truly believing in science are, I think, a little incompatible, that oxidative damage was the cause of aging. And he was very effective in pushing this, and it's a very attractive hypothesis. But we now know that it may contribute to this thing that we've talked about by aging, but it also, in certain aspects, can help. You know, a small amount of oxidative damage may actually be important or beneficial to preventing some of the aspects of aging. Related to that is mitochondrial damage and dysfunction. I'm not going to go over these in any further detail, but I just wanted to give you a flavor. And many of you probably, some of you may have already thought about, well, I think aging is caused by this or that. And you may be right. But the big question is, how do you test that hypothesis? How do you test the hypothesis that any of these things are involved in aging? Does anybody have any ideas how you would do it based on what we've talked about so far? Some of you, yeah, go ahead.
You artificially increase one of those factors and measure the rate of disease over a long-term population.
And what would you measure?
The rate of death, or maybe incidence of disease in some susceptible mouse models.
Exactly. Yes. And the question then becomes, because here's how it was done before people started thinking like you. They just described these changes in young, middle-aged, and older organisms. And indeed, when they did that, they found, for example, that oxidative damage to DNA, to proteins, to certain lipid moieties increased with advancing age. But you know, that's just a correlation. My hair fell out and my hair has gotten gray, but is that causing me to be more likely to die from a bus hitting me than something else is? It's unlikely that that's going to be causing me to have these really problems during aging that we're particularly interested in. I mean, I'd like to get my hair back, I think, but I'm not sure. But I'd really like to not have this doubling of mortality with every 8 years as we get older. So there was a lot of stuff, and what really enabled people to make and hang on to these hypotheses initially was descriptive biology, showing that yes, telomeres shorten with advancing age. There's more DNA damage with advancing age. There's mitochondrial damage. Many cells, including stem cells, are more likely to stop dividing with advancing age. But the problem is, unless you can manipulate that, unless you can have an intervention to change that in some way and then measure the effect it has on aging, you won't be able to get anywhere. So, for example, mTOR signaling is a recent hypothesis about a cause of aging. mTOR, as you know, is a part of the nutrient sensing pathway, and it regulates cell growth, cell division, and people started thinking, cell growth, cell division, cell senescence, maybe this central signaling pathway could be a factor. And the question then became, how can we find out? Okay, so intervention is the tool. And everybody likes to think of pills as a way of solving any problem. And so people started thinking about ways of finding interventions that would manipulate these various hypothesized causes of aging and would slow down the process. Okay, ideally, you'd even like to be able to administer a pill when you're old and reverse the process. That would be great. And in fact, there's some evidence that there's proof of principle that certain aspects of aging can be reversed. This is pretty late breaking data. And there's definitely proof of principle that certain aspects of aging can be prevented, or I should say slowed down. Okay, so intervention serves two roles, actually. It serves the role of hypothesis testing, as we just described. It also it has a translational potential that if you find a drug that, say, interferes with mTOR signaling, which is hypothesized to be involved in aging, but then you also, and you find it does, you've already got potentially a drug therapy for some aspect of aging. And, there are lots of drug therapies being advertised out there right now. Amazing, it's a multi-million, I would hazard to guess, $1 billion business for human growth hormone, testosterone therapy. Many kinds of nutraceuticals are out there. Some of them are deleterious, actually. The growth hormone story, for example, is a good example. People reasoned, this is a good example, that because growth hormone levels are known to decline with advancing age, well, anything like that changes growth hormone is anabolic, muscle mass, so on and so forth. We should jack up growth hormone in old individuals. And so they started doing it, and people are still doing it without the evidence to support the idea that it is beneficial. In fact, it has a lot of deleterious side effects. It leads to carpal tunnel syndrome in elderly people who are taking it. It increases their risk for diabetes. Growth hormone is an insulin desensitizer. It has many other deleterious effects. So you have to be careful. And that's what led the National Institute on Aging, which is one of the ironically youngest institutes at the National Institute of Health around. It started just when I was a graduate student working with Dr. Finch. That's why the National Institute on Aging set up what they call the intervention testing program. And so the idea was twofold. It would allow us to test hypotheses about what causes aging, and it would allow us to test drugs that were already out there on the market to see whether or not they were beneficial as they'd been hypothesized to be, or even deleterious, okay? And so what I'm gonna go through now is some examples of this, but I wanna tell you already in advance two interesting things, and there's some lessons to that. 12 years ago, when the NIA put out an announcement to everybody in the United States who wanted to compete for it, that you could compete for being one of the interventional testing program centers in the United States, my personal thought was this was a boondoggle. It was just a waste of time. And it was based on not much evidence. I was imposing onto this idea my own feelings about it without thinking a lot about the evidence. The evidence out there was that people like Dr. Harmon, the guy who was always the true believer of the free radical theory of aging, he, for example, had administered antioxidants to mice as they throughout their lifespan, and there was no effect. He said, well, I just didn't give high enough a dose. He wanted to believe this. But there were other papers in the literature that had tried certain drugs, all to no avail. Okay, so that was my justification for being skeptical about it. Okay, so we actually still wrote the grant, and we were fortunate enough to be one of the 3 testing centers. The other one is at the Jackson Labs in Bar Harbor, Maine, and the third one is at the University of Michigan. The idea was to have 3 sites so that you would replicate what was done. Replication is so important. I should tell you from my perspective in the aging field, there must be 20 papers at least in Science and Nature and other leading journals that report either a genetic manipulation, usually it's a genetic manipulation, a knockout mouse, a transgenic mouse, that has extended lifespan. Only 1 or 2 of those papers has ever been reproduced. And I don't mean somebody's tried to reproduce it and failed, but just, I mean, they may have, we often don't know about those things, but then there's been a publication out there to show that this occurs at another time. And that's really important. So having this replication is very important. But I was wrong. I was completely wrong. In the first, every year about 3 to 5 drugs are entered into this program. Anybody, even yourselves, student, a lay person, or a scientist, can submit a 2-page proposal. Dr. Walzem, this is how I met Dr. Walzem, we're on the initial review panel of these proposals. She was brought in because of her nutritional background and her knowledge about nutraceuticals and things like that. We review these proposals, and all you have to do is have a good argument that compound X was shown in an invertebrate model to extend lifespan, or there was some data where we administered compound Y to mice and it reduced their oxidative damage, and it reduced the incidence of cancer in these mice, and it also reduced the cataracts they had. The bigger case you can make for compound X, Y, or Z, the more likely it's going to be thrown into this program. And I'll tell you, there have been some compounds thrown into the program that were on very shaky grounds, and some that weren't. And I'm not going to go too much farther down that path, except to say that of the first 14 or 15 compounds that have been tested, one interesting thing is none of them were deleterious in terms of the aging measures that we measured. None of them shortened lifespan, for example. That's pretty amazing. Toxicology, you might think that these drugs might, some of them are known to have certain well, anything will have toxic effects at a given dose. The other thing that was even more remarkable is 4 of those first 12 to 15 compounds extended lifespan in at least one of the sexes. 2 sexes are studied. That's an important point also to make, is the model that's being studied. It's a heterogeneous, genetically heterogeneous model. We don't study it in a single inbred strain of mouse. We study it in a cross between 4 inbred strains of mice that on a diagram of distance, genetic distance, you might say, they are more distant from each other than any other set of inbred strains around. So they're genetically heterogeneous just like we are. So that in the end, if you find one of these drugs extends lifespan, you can't be worried about the fact that, oh, it's doing it in a C57 Black 6J mouse, which has certain genetic tendencies to develop specific cancers and die from them, to have certain types of sarcopenia, et cetera, et cetera. You're only affecting this one strain. So it's the reason for genetic heterogeneity. So this is a restatement of what I already told you about the reasons for this program. So we'll skip that. But then you've got the big issue, which I talked about at the beginning. What is aging? What are you going to measure? Okay, what criteria in these studies where you administer a given drug are you going to measure in these mice to establish whether or not it really is slowing down the aging process. You had already given an example, but before that, you have to, first of all, show that the intervention you're using is altering the postulated mechanism. You have to demonstrate in your animal model that what you're measuring is affecting the mechanism that you're hypothesizing it to work through. The intervention must be shown to extend mean and maximum lifespan of the population, and we'll define those in just a minute. Obviously, if it's affecting aging, broadly speaking, it's going to turn these mice from normal mice into centenarian mice. Centenarians, for example, is another way of studying aging. Centenarians are operationally defined as humans that live up to and beyond 100 years. Sometimes people use 95 as a cutoff point. And it's an interesting model because usually centenarians, when you look at them at 80 years of age, they look a lot younger than their peers who aren't going to become centenarians. That's a whole area of interesting study. But anyway, your intervention is going to be required to extend lifespan. But as that's not enough, okay? Because it's possible to extend lifespan without delaying and reducing all age-related diseases. For example, you could have the C57BL6J mouse. About 70%, 80% of the mice, when they die, you find them filled with lymphoma. They're very susceptible to lymphoma. So if you stopped with an intervention at the point of just measuring mean and maximum lifespan, and you found out that these mice were cured from having any lymphoma, you would extend the lifespan of those mice. But would you have affected aging? Not unless you established that this mouse also had delayed or reductions in all of the other diseases of aging. And even that would not be enough. You'd want to also be sure that it delayed and reduced all the deteriorative age-related changes that we talked about, such as endurance, such as muscle strength, cataracts, anything that would balance cognitive function, kidney function. But then where you stop, okay? And so that's one of the problems that this intervention testing program is faced with. It can't afford to do all of these things. The program is funded now at the level of $1 million per site, but the animal care costs, the costs of measuring all of those things far exceed those budgets. So you have to step back, and initially the criterion is the drug has to extend mean and maximum lifespan, and you have to get some evidence that it hasn't just affected one disease of aging. The idea is that if you find that much and it's repeated at 3 different sites, you've set the stage for saying this intervention is a candidate for delaying the aging process, okay? And then people can start exploring all of these other aspects and see how broadly anti-aging it is. Okay, so the one we're going to go through a little bit more detail as an example is Rapamycin. And I'm showing Randy Strong, who's the head of the intervention testing program at San Antonio, and Dave Sharp, who submitted that 2-page proposal with the argument that convinced the review panel and everybody else who followed suit after that initial review that it was a worthy candidate for an intervention. Okay, so it met criterion. Rapamycin suppresses mTOR activity, and that was the hypothesis, that mTOR activity plays a role in limiting lifespan. Yes?
You were talking about this argument, but wouldn't the combination treatment not fulfill this criteria of an anti-aging intervention?
You mean like rapamycin plus something else?
A cocktail or nutrition is actually a combination treatment.
Exactly. I think that I don't recall the panel ever being offered that suggestion.
You know, the thing is though, that there's so many pharmacologists on that, they really like one drug, I mean, I have picked that up. That, you know, we want to test them sort of before we start combining things. It's been a little bit more pharmacology rather than, as you say, nutrition with a spectrum, a cocktail.
But, you know, if you have an argument,
Yeah, if
if you have an argument, like I can start imagining things that you have data showing that drug, nutraceutical X and drug Y alone affect these endpoints and together are synergistic, let's say.
Because nutrition has sometimes many targets at the same time.
Yes, absolutely. So I think, you know, it's an important point. I think you can make a case for it. And I will tell you this, that, you know, this program's been going on for 11 years or so. And in process now, 2 of the drugs are being studied now in combination. Metformin and rapamycin.
Right.
Okay.
Yeah.
So it's starting, you know. We just have to hope we can hang on long enough to keep, you know, these things going.
Well, we had those guys with Protandim and things like that that were not really defined.
Yeah.
They were just a thing that they thought was, you know, I mean, they had a mechanism then that was like, we don't really know what's in there.
Yeah.
Even in medicine, you see that the real advantage and that people live longer or have less mortality from disease has not been caused by one intervention. It's the combination of many different types of interventions that at the end led to a higher survival for a lot of disease.
Exactly. As long as you don't get into polypharmacy. But no, I think your point is very well taken and a very good one. And you've, in some ways, anticipated what's already starting to happen. Okay. Okay, so it meets criterion, but you know, you have to test your animal model to be sure. We know mTOR activity can be reduced by rapamycin from all kinds of evidence, in vivo evidence, in vitro evidence. We had to test in the model that we're studying that the dose of rapamycin administered to the model had those effects. That's absolutely essential. And then you ask, does it extend mean and maximum lifespan? Now, what do I mean by mean and maximum lifespan? I wanted to spend a little time on that because it's really, I think, an important tenet of aging research. And it can be studied in a more rigorous mathematical way than this, but this is just to give you a flavor for what we're talking about. I'm sure you know what median lifespan is. It's when half of the population that you're studying, half of the population that you're studying has died, okay? So that's median lifespan. Now, as you can see here in these 2 examples of lifespan extension, you can, and of course there are many other cases, but you could have a case where median lifespan is extended, but maximum lifespan, and usually that's defined not at the age of the last animal to survive in the population, because that's an N of 1, and I'm sure you've all had enough statistics to know that that's a very unreliable sample. So if you have a large enough sample, you can sometimes get statistical power by looking at the age at which 10% of the population has died. And so in that case, you can see in this particular population, none of them live any longer, none of them survive to the 90th percentile any better than the controls do. But you have this other option or alternative where you have both median and 90% survival extended. And this kind of a pattern has long been touted as being a better indicator of an intervention that affects aging then this kind of pattern. That's not to say that this isn't valuable information. Anything that would enable a population, presumably human, to have a larger fraction of them live out to 50% survival probably would be a good thing, okay? But it's very likely that in that case of the C57-by6J mouse in which you eliminated all lymphoma, you would see this pattern. But once you got out to here, when the animals were suffering from all of the effects of senescence even more than just dying from lymphoma, you wouldn't see any difference in survival. However, if all of the factors that affected mortality were affected by the intervention, you would more likely see this pattern. So that's why the criterion is that you have to have success in extending median and maximum lifespan to really feel that you're having an impact on aging. And again, that's only first criterion. Okay, so let's look at rapamycin. Rapamycin has now been studied in a number of iterations. First, as I said, it was studied at all 3 sites, and we're only looking at the males. I should tell you the females on rapamycin do have greater extensions of their survivorship curves than the males do. But so I want to look at the males first. I'm not going to show you the females, but I just wanted you to know that. And what we've done here is the first study was done at this dose because this dose simulated the human dose. Rapamycin is clinically approved for treatment with transplants, like kidney transplants, as a immunosuppressive agent, actually, in concert with a number of other drugs. It's also being in trials for kidney carcinoma. It suppresses mTOR. So that was the dose that's used in humans, and that was the guess that was made for this trial. And rather amazingly, as seen by the orange pattern, this was a significant extension in mean, and believe it or not, you can't see it, but the maximum lifespan at 10% was also statistically significant. This is pooled data across the 3 sites, so the sample sizes here are quite large. They're in the neighborhood of 120, something of that nature. Don't quote me on it, but that's a big sample size for mouse studies because they're so costly. And then you can see that at the higher dose, it's even more effective in extending median lifespan. So the big question I would ask you, though, and everybody needs to ask, is whether or not it's really meeting that second criterion, even though 10% survival is greater. And I want to put a comparison here. This is dietary restriction, a study that we've just completed on the same animal model, that genetically heterogeneous model. And what you can see is there's no question that maximum survival has been affected by dietary restriction as compared to what's going on with rapamycin. So under these circumstances, DR is certainly trumping rapamycin as a factor of survival. So bear that in mind. So it extends lifespan and maybe extends maximum lifespan. That's somewhat questionable. Does it delay or reduce all diseases of aging? Well, now we're moving into a grayer area because the detailed pathological assessments in this particular model have not been that thorough. And so I'm just going to give you some of the rough data out here that I think is pretty reliable. Number one, it delays cancer, but it only delays some of the spontaneous cancers in these animals. It may retard aspects of cardiac aging. We know it impairs reproductive function, one of the age-related changes that occurs in both men and women or any animal model. as they age is that reproductive function at many different levels declines. Well, this causes testicular atrophy, for example. It also, there's some less strong, but not as, because it's been studied as much evidence that ovarian follicular growth is also impaired by rapamycin. It increases cataract formation. That's going in the opposite direction. It enhances some resistance to pathogens. That's very interesting because many people argued, well, rapamycin is used to suppress the immune system. Quite interestingly, the data in older mice indicates that it doesn't suppress any resistance to pathogens that have been tested so far, and it's a limited number. And it actually increases resistance to some pathogens. But it also reduces insulin sensitivity and glucose tolerance. And that's not always considered, I mean, that can be a negative effect. So it's a mixed bag at this level. So it only reduces some of the diseases of aging. And then the next question is, what about those multiple other things that we want to control if we're influencing aging in a positive way, the deteriorative age-related changes? Now it gets even messier because there's two big articles out there. One that was published from San Antonio Group, the first one, the second one, and another one which is much more comprehensive in the number of measures that have been made. Remember, I told you we can measure ad infinitum age-related changes. And that one argues that it extends murine lifespan, but it has limited effects on aging. I don't have time to go into all of the details of that, but I'm just showing you how, in its infancy, this approach to studying aging is. The vast majority of things that have been looked at with respect to aging, be it in C. elegans, Drosophila, mice, have been lifespan, much less in terms of diseases and far, far less in terms of life and health span. We can follow this up later in questions if you have any questions, or at lunch with the students, because I want to go on here. Okay, so I just want to give you a flavor for some of the other drugs that so far have been found in this program to extend lifespan. NDGA, nor-dihydroguairidic acid, which is a component of the creosote bush, which is the signature bush of the Chihuahuan Desert, and Indians first made tea out of it, out of the creosote, and they didn't know, of course, they had NDGA in it. It's also used as an insulin sensitizer. Aspirin, both of these worked in males only and have been repeated now in a second experiment at a higher dose and still only worked in males. Rapamycin, 17-alpha estradiol, and a relatively inactive estrogen. 17-beta is the active estrogen. And finally, acar, which is a non-absorbable polysaccharide, I think, that that has been used as an insulin sensitizer in European clinical studies treatment for a long time. And I think I'll show you some data on that momentarily. And those are some of the references on it. Yes, here is the data for rapamycin and acarbose. And you can see another interesting aspect about these studies is that there usually is a sexual dimorphism or differential response to the drug depending on sex. As I had told you before, females generally are responding with greater increases in mean and maximum lifespan than the males are. But quite the opposite of this is the case for acarbose, where you get this quite dramatic response. It looks very good for males and minimal but statistically significant for females. And now I want to show something that I think is very interesting, which is to put food restriction from the same animals, not done at the same time, but this food restriction work has been repeated by Dave Harrison, and that's already in San Antonio. What I'm doing here is scaling these graphs because I didn't scale them to exactly the same metric. They're listed up here in months, and they're listed here in days, but what's 24 months on this scale is 2 years in days on this scale. So what I wanted you to see was how effective these interventions in comparison to what you might consider the gold standard, food restriction. And if you look at the males, 50% median lifespan under dietary restriction is barely reached under rapamycin under any of these circumstances as it is by acarbose. Even though maximum lifespan is increased by acarbose, it's nothing in comparison to what's achieved with dietary restriction at 20% survival. Okay, so there's some interesting hypotheses that can be raised about mechanism here, but they remain to be tested. And similarly, in females, there's a larger response to dietary restriction, with one exception. I think median lifespan under rapamycin at the doses used can be as effective as dietary restriction in females. And then look at this other interesting result. Remains to be replicated, but it's not uncommon in food restriction studies to see that there's a subfraction of individuals who actually have early mortality under dietary restriction. And there are other models that show that as well. Okay. Okay, so further comparison of dietary restriction to these interventions in terms of meeting criterion, if you go down the list where dietary restriction has been around long enough and studied by enough people with as many different levels of expertise to show that it does indeed delay and reduce most diseases of aging. It doesn't cause cataracts, for example. It can suppress reproductive function at high enough levels. And only when reinstituted is reproductive function restored, and actually restored to a better level than age-matched controls. So it's not completely anti-aging, even in terms of diseases, and there are others as well. And it delays and reduces only some deteriorative age-related changes. One of the best examples of that, I think, is expression profiling data, where you take tissues from an old animal and compare them to tissues from a young animal in terms of the level of expression using Illumina or any other arrays possible, the changes in genes expression as a function of age. And there are thousands of changes in the liver, for example, thousands of genes whose expression levels change with age. And then you do the same thing with food restriction, and you find out that food restriction only reverses about 30% of those changes. And the others are either not affected or in some cases go in the opposite direction. So is dietary restriction totally anti-aging? That really raises the question, I think. Okay, so the last thing I want to do is just talk a little bit more about this work on dietary restriction, showing the genetic variation as an example of genetic variation in response to interventions. This work, as I said, was done and thought up by these individuals. Okay, and these are just two examples from the 40 strains, inbred strains that we looked at. And this data is pooled for males and females. But when we started getting this data, for example, look, DR is red, dietary restriction. Look at this strain. We started seeing animals die under dietary restriction at a very early age. And initially we thought, oh, it'll cross over, just like I showed you for the female, but it never did. And then we saw a large number of animals who didn't have any effect, and we saw animals that had this effect. This was very hard to publish. This is just the recombinant inbred series. I don't have time to go over that now. And this is the model in which we looked at 41 strains, males and females. We restricted them to 40% of their cognate strain food intake and followed them throughout life. And this is what we found. First of all, under ad lib conditions, the inbred strains had this remarkable variation in, we're looking just at mean lifespan here, because our sample sizes weren't big enough to look at maximum lifespan. And then, as you can see, under the red bars are for the strains in which dietary restriction increased lifespan. And that's the C57 lifespan equivalent, which was a lifespan under ad lib conditions midway between the lifespans of the shortest and longest lived under ad lib conditions. And when you plot it from ranking them according to the biggest effect on lifespan positively to the biggest effect of lifespan negatively, you see it's a rather continuous line of effect, showing that there's just a lot of genetic variation in the response to this intervention. This has never been done for rapamycin, for any of the other interventions. I would hypothesize that the same thing would be achieved. And this can serve several purposes. You can go back and start studying these animals who have, these strains who have shortened lifespan and identify genetically and physiologically what's different about them from these strains. And we've begun to do that, and I can tell you about that if you have questions. Furthermore, an example of what you can do is looking at the same thing with yeast. This is work it's now been published since I made this slide by Matt Haberlin and his group at University of Washington, where they screened many different yeast, and you can food restrict yeast by diluting their medium to a reduced level of nutrients, you might say. And they saw basically the same thing with a much larger panel of yeast, going from percent change in lifespan being negative all the way up to positive. And each of these lines, they knew what gene had been knocked out in these lines. And as proof of principle, to show you what their initial bioinformatics revealed was that there was only one grouping in which lifespan was lengthened, and it was a grouping in which certain aspects of mitochondrial compartment their genes were interfered with. Things that would lead to shortening of lifespan was a whole host of pathways and compartments as shown here. And I'm only going to say that this shows you the power of this kind of approach to getting at mechanisms that limit the effectiveness of a drug or an intervention like food restriction, and can lead to many, many new and interesting hypotheses. Here's another example of a negative effect. What we're looking at here in food restriction is mice in which the gene regulating corticotrophin releasing hormone, which is elaborated in the hypothalamus and regulates the whole hypothalamic pituitary stress axis through the adrenal secretion of corticosterone. And what you can see is the red the red line is knocked out animals not having this gene and not being able to elaborate glucocorticoids under food restriction. Their lifespan is markedly shortened under dietary restriction. If they have the knockout under ad lib conditions, shown in yellow, the knockout doesn't affect lifespan markedly at all. With larger sample sizes, it reduces it by about 5%. So here's an animal without the gene regulating the hypothalamic pituitary stress axis that just can't handle food restriction and has a shortened lifespan. It's just another example of the power of manipulating the genome to get at this. So in conclusion, this is one person's view of how calorie restriction works, and it remains to be tested in ways that the gentleman who answered my question at the beginning first mentioned, that you have to intervene in all of these pathways, all of which have been shown to change with aging, but you have to intervene in them and see whether or not you lose the benefits of calorie restriction when you reverse what calorie restriction does to any one of those pathways. That's point number one. Point number two is remember rapamycin affecting mTOR and how its effects, in terms of longevity at least, are a fraction of the efficacy seen when you calorie restrict the animals. Maybe this is part of the reason. Affecting mTOR is just one of the pathways, many pathways hypothesized to be affected by calorie restriction to give increased lifespan. And this actually gets to your point about finding a combination of interventions that can be effective as dietary restriction. Okay, so in summary, what I hope I've shown you is just the problem that we're faced with and a little bit about how to handle the problem. Defining aging, how difficult it is to do, how complex and expensive it is to do in animal models. Interventions that modulate aging were only at the very beginning, but we're way far along compared to when I started this 30 some odd years ago, where the only intervention was dietary restriction. And back then we didn't even know how broadly effective it was. And the implications for these interventions have for identifying mechanisms that underlie aging. So thanks for the opportunity to talk to you, and I'll look forward to meeting you and talking with some of the students at lunchtime.
We're running a little late. We only have time for maybe a couple of questions. Does there somebody have a burning question?
I apologize for coming in late, but I missed whether, when you're speaking of dietary restriction, is it food restriction or only calorie restriction? And what, for example, are protein levels used?
Yeah, I'm shaking in my boots in the nutrition department. It's food restriction.
Okay.
In fact, that's why we call it dietary restriction.
Yeah.
Just a very interesting story about this that I told some of the others. Initially, when Dr. Masrio, who was the chairman of physiology and one of the pioneers in starting back food restriction back up, it was discovered in the 30s. But in the 70s, when looking at DNA was formed, he realized that this was the only intervention that affected longevity, and therefore the only thing worth he felt studying. He talked to nutritionists, and they said you have to have a defined diet. So he did, and it was casein, protein, I forget what the carbohydrate was and the fat was. It was defined. So he had this huge effect. It was Fisher 344 rats. It was like bigger than anything I even showed you with the mice. 40% increase in lifespan. He noticed that all of the ad-lib rats were suffering from terrible renal nephropathy. It's a characteristic of the Fisher 344. But as they got older, and he started talking to pathologists, and they said casein is not good for the kidney. So he changed it to soy protein, and he increased the ad-lib lifespan by 15%, or even more than that. The soy protein did nothing for the food-restricted animals. It's totally survivorship. So then people got lazy later on as things opened up and VR was not being challenged and people avoided the journal of nutrition and so on and so forth. And they just started doing dietary restriction. And in fact, we did this with a Fisher 344 rat and we fed them green rat shell, food restriction. Same amount, 40% restriction. On green rat shell, the ad-lib animals did even better in survival than they did under soy, and under food restriction, the lifespan of the food restricted animals didn't increase one iota. So if Dr. Maslow had started his study without taking the advice of the nutritionist, he would have gotten a result of about 15%, and he might have quit. Okay, so the nutritionists were very helpful. And yeah. So it's a very good question, and there's a whole, we were just talking about this, there's so much to explore in terms of nutritional components.
One last question.
It seems that this observation of the short lifespan really puts the onus on the physiologists and people who are determining cause of death. And then, of course, later these experiments they can really get down to. So the flip side question can be asked, is something which extends life, showing all these effects on the diseases of aging and also on the physiological stuff, and also when life is shortened by these things, is it because one particular pathology then crops up, or is it still very generic in the sense of what do they die from it, and in what ways do they decline?
Yeah, and that's an important question that's being followed up, I know, by some people who are asking, maybe not quite the same way, because it's really hard often to identify, particularly when a young mouse dies, why it dies. These animals aren't looking like they're starving or showing signs of nutritional decrement that they might be. But the question is, these animals may be showing all the benefits of dietary restriction, like I didn't tell you, they're hugely resistant to toxin stresses. Most toxins, the LD50s, go up dramatically. Maybe they still have that resistance, but something else is killing them that they can't have. One thing they do is their glucocorticoid levels, the ones that everybody in that series, glucocorticoid levels shoot up. This is universal so far.
Whether or not they live short or long.
Yeah, but they shoot up a hell of a lot more, again, the ones that live shorter. And also they lose adiposity to a great extent. Some of the ones that benefited, to our amazement, this fat reduction was also a big argument for effective food restriction. Some of them actually don't show any loss proportionately of their fat mass.